So you should be able to start sharing your screen now there, Peri. Peri is a plant scientist at the University of Sydney. She is broadly interested in the complexity of plant molecular responses to pathogens. She completed her honours in agricultural chemistry and PhD at the University of Sydney after a career in horticulture, arboriculture and environmental management, and raising 2 children. She has secured several grants, including a recently awarded ARC linkage grant with plant and food research here in New Zealand to investigate Myrtle Rust. Peri, I believe you should now be able to commence your presentation.
Thank you, Renee. I'm hopeful that you can all see my screen.
I can see it, so there's a start.
Okay. All right, so I thought today, I've talked a lot about the genome, and I just thought today maybe I'd give a little bit of background into how and why we went about building this genome for Austropuccinia psidii, and just also what we hope to achieve with it. And so that I think I'll just refer to it as Myrtle Rust, though the actual name is Austropuccinia psidii for this organism. And this picture here is just of some spores that the nuclei are fluoresced with propydium iodide. So you can see that it's got a dihaploid nucleus within a single spore, which is kind of the state of this spore, this rust fungus. I won't go into a huge detail on the biology because this whole seminar series is on Myrtle Rust, so I'm presuming a level of understanding that that most people who are listening will have. But just a brief slide just to show the infection on a leaf of how this affects a leaf. This is an Agonis flexuosa. It's a West Australian myrtle plant, and it's growing up the road from my house, and you can see that it's infected. Just this is field infected. And the spores from those pustules, you get masses of spores, and they're all released into the wind, and they drift onto another plant and parasitize the next host. So they're quite damaging and spread quite rapidly. In 2013, this map came out from Criticus et al. Based on some climate modelling and where predicted spread of Myrtle Rust would go. And you can see globally where this pandemic was likely to spread from those maps. There's been a few other iterations of maps since then, but this year a publication came out and this looks at where Myrtle Rust has been detected, or the presence data is available, and you can see it doesn't exactly match the map, and that's probably due to field survey work and highly intensive in Australia and New Zealand, and perhaps not so intensive in some other regions. But I think we can kind of feel that this is a pandemic. This is really spread. Carnegie, so Angus Carnegie and Jeff Pegg published these really nice graphics to kind of demonstrate the spread of the Myrtle Rust disease, and we can see that as there are several biotypes, so there's several strains of the rust fungus, and the pandemic, what's known as the pandemic, which is suitably coined because of its wide spread and wide host range, has really taken off since 1977, with presence now right across the Oceania Pacific region and other countries. South Africa seems to have another biotype there, and I guess they're very worried about getting the pandemic strain there as well. But yeah, Australia, 2010, we saw the first detection of the pathogen, and by 2011 was deemed ineradicable. And New Zealand, of course, it's well known over there that it was 2017 when it arrived. So in 2010, when we first detected Myrtle Rust in Australia, there was great alarm and quite a lot of research and a bit of funding to try and do some analysis of this. And I guess initially we realised that there was no genome for this fungus, and to start to understand the pathogen and how it's manipulating the host, it's really useful to have a genome so that you can start looking at molecular responses, and that was a certainly decided that that was something that we needed to do. And so there was some funding for that. Mu Keng Tan and her team assembled the genome for the pandemic strain that she got the DNA from the Plant Breeding Institute from Robert Park, who'd already determined that across a variety of rust that had been detected in Australia, that we only had the pandemic. So they assembled the genome. It came out at 103 to 145 megabase pairs, and they used the state-of-the-art, I suppose, sequencing at the time, which was using short-read sequence data. And that estimation, now we know, obviously, is incorrect. But they did determine a large proportion of transposable elements, around 27% of the genome, and they also placed the phylogeny, Austropuccinia psidii, on the separate Pucciniaceae lineage. And you can see that their estimation, so I'm just going to get a highlighter. You can see that their estimation for the genome size was sort of reasonable. It was in the ballpark for Pucciniales, so most of the genomes are sort of that we know of around less than 300 megabase pairs, although there are outliers. So it kind of made sense, and it seemed that there was something with the repetitive nature of the genome that was meaning that so when it was being assembled, it was a bit collapsed. So Robert Park, who supervised my PhD and has had a very long-standing interest in Myrtle Rust, in 1983, he tells me he applied to go and do a postdoc in Brazil to study Myrtle Rust amongst other fungal leaf parasites of eucalypt. So it's been known that this is a looming problem for Australia and New Zealand for quite some time. And I guess it's just been a long time coming that we've actually found this disease here. So following the short read assembly, which I've just mentioned, we realised that wasn't going to be suitable for much of the work we wanted to do with the rust to understand its pathogenicity. So Robert Park initiated in 2016 a PacBio long read assembly project, and Dr. Chong Mei Dong did some fantastic high molecular weight DNA extractions and sequenced that DNA down in Adelaide and also here in Sydney on the Ramachiotti machines. At that stage, we were, this is kind of technical, but we were dealing with the early generation of long read sequencing machines, which was the RS2. And we got 18 smart cells sequenced. It didn't assemble well. Later we realised we only had around 12 times coverage, so that's probably why it didn't assemble very well. I just finished my PhD, and I was looking at RNA sequencing in resistant versus susceptible plants in response to Myrtle Rust, and then Robert sort of suggested maybe I could try and find out what the genome size is using some flow cytometry and KMA analysis. And so that's what I did. I did a KMA analysis using the short reads that were available, and it seemed to come out at around 1 gigabase, which I thought, well, maybe I'm doing something wrong here. And then I did the flow cytometry using infected eucalyptus leaves, and that came out at 1.4 gig. So we sort of think at this point, right, we're dealing with something a bit bigger. And so we sequenced 3 more PacBio cells, and we got 36 times coverage. So this time we're on the next generation of PacBio sequel. So, and I assembled that. And so the first assembly results were in 2017 using that 36 times coverage using long reads. And using haploid parameters, so this is just collapsing to a haploid genome, I got around 1.4 gig genome, 76.7% complete. It took around 2 months to run continuously on a high performance computer. And then I did diploid parameters. I got a 1.5 gig genome and 79.9% complete. So we're getting a little bit better each time, but still not, still missing quite a few conserved genes. It's not there. I then, in 2017, Myrtle Rust was detected in New Zealand, and I then met Grant Smith at a conference, who gave a fantastic presentation in Canberra and suggested that the New Zealanders were going to do a long-read assembly of the genome. And I also at that time met Benjamin Schwesinger at ANU, and we got together, all of us, and decided to join forces and really make a much better job of this, pool our resources. So David Chonier and Grant and Benjamin and I and others, and many others, I should say, joined forces. We sequenced a further 8 SMART cells, and we got some high-C data and RNA-seq to try and phase the genome and to annotate it. And just early in 2018, a publication came out of a draft assembly of the South African biotype, and that estimated the genome around 1.2 gigs. So that was kind of adding more confidence to my initial predictions. And this is a slide just to show that the assembly happened over 5 months, at any time using 480 gigabytes of RAM, 120 CPUs, and we built a highly contiguous genome with the latest chemistry, sequence technology, and software at the time. I like to just state, because things change so fast. So this is just to compare. This was my previous diploid assembly using 36 times, and this is we had 72 times coverage for the final assembly, and we got a final assembly of nearly 2 gig. That's the 2 haploid nuclei, right? And it took 4.5 months, by the way, continuously running, and 89% complete. So it's looking a lot better. We found the GC. I then duplicated the genome so that we had 2 separate haploid genomes, primary and secondary. And with Chen Wu and Cecilia Deng in New Zealand, we managed to do some scaffolding using the high-C data. We came out with 66 final scaffolds and a pretty good N50 and L50, for those that know what that means. And the primary genome assembly was 1.018 and a very low GC content. So why has it got such a low GC content, and what are some of the characteristics? Well, when we did a little bit of downstream analysis on the genome, we found that the gene spaces, that is the gene lengths of Austropuccinia psidii, which are the red bars, were similar to other rust species, but the intergenic spaces between the genes were like an order of magnitude different. So we could see that there was very, very big gaps between the genes. And what we found was that those gaps were largely transposable elements, largely from the gypsy LTRs, and they covered more than 90% of our genome. So these transposable elements are kind of regions of DNA which are, as the name says, transposable. They jump around, jumping genes, as Barbara McClintock kind of first identified. And this is what we're seeing in our genome. We also found extended telomeres compared to other fungi. So, and again, an order of magnitude longer. So we found 29 telomeric regions. 10 of those post-scaffolding are greater than 1,000 base pairs. Sorry, 1,000, not base pairs, 1,000 copies of this repeat. So really, really large. Comparing the overall GC content with other rusts, we can see that the Myrtle Rust has a peak at around 33%, and then another peak around 40, just over 40%. And so we now have, compared to other rust fungi, this is like, these ones are more like 45, 45, 45. We're really in a different ballpark here. So that seems to be really very clearly due to these intergenic transposable element regions. And we did some analysis of those which are AT rich. So these, so what happens with these transposable elements over time is that they get methylated, and then they are there's a cytosine transition to, so you get a CPG transition to AT, and this just happens biochemically over time to methylated regions. So what we see is older, on these plots, we've got like the older transposable elements on the left and the younger ones on the right, and you can see that over time there's a depletion of GC to AT. Overall proteome conservation, so when we looked at the ortho group, we used OrthoFinder software to look at protein ortho groups, and there's a conservation of quite a large number of genes. We can see there are 633 proteins that are Myrtle Rust specific, 300, and there's 3,843 ortho groups which are present in all the species, all these species that we looked at. And interestingly, 101 ortholog groups are only present in tree rust, so that might be something worth looking at later. When we look at the phylogenetic output from OrthoFinder, we can see that the Myrtle Rust doesn't cluster with the tree rust, but it's kind of out there on its own. So the key findings were that we had a very large contiguous genome assembly bloated with transposable elements, and there are clear TE bursts in recent history which contributed to the genome expansion. There's no evidence of repeat-induced point mutation machinery, and this is kind of interesting because we think that largely this is clonally reproducing, and the repeat-induced point mutation machinery, I believe, is only activated during meiosis. We found around 19,000 coding genes, around 367 predicted effectors, and there was 97.6% conserved proteins, very large telomeres. And the proposal that we've made in our paper is that we propose that the recent TE burst occurred in a small subpopulation and was beneficial to host range expansion and adaptation, and it kind of fits with the radiation of the Myrtaceae. The publication pre-proofing is out on this website, and it's also available here along with some annotation files and Effector, the putative effectors and the transcriptomes are all available at these sites, and I think Renee will share those later. So what we're doing now is doing some downstream work from that and looking at comparing the total predicted proteins across biotypes of the rust. And when we look at an analysis of those, of a Brazilian isolate compared to South African isolate and our one in Australia, we can see the total predicted proteins are fairly conserved or fairly similar. But with when we look at just the effector proteins, we can see some variation here. And when I mapped the RNA from those 3 different biotypes to our pandemic genome, this is one of the putative effectors. So these are the genes that manipulate the host to parasitize. This is shows some SNPs and shows some variations across these 2. So this is the RNA mapped, so for the South African one, mapped to the pandemic genome, mapped to the pandemic genome. This is the Brazilian, and this is obviously the pandemic RNA mapped to the genome, so it matches fairly well. So we are seeing some change, some variation there. We've also been testing a few things in the lab, and one is testing nanoparticle delivery of ribonuclear proteins with the CRISPR-Cas9 for gene editing. And we're having a little bit of success with that being transported into the germinated hyphae. And this is just a really proof of concept, and this wouldn't leave the lab. This is really just the idea with this is perhaps that we can test some effectors, you know, to validate them. So this is kind of not to be released. I'm coming up to close to time, but I'm just going to quickly go through some. We know that there are some problems with the genome. So scaffolding was problematic with the high C, and we know that now looking at these interaction frequency maps that we have some chimeras. So we know that there are issues with the genome still, and that was probably due to the generation of the sequence reads, as well as a few other problems that we had with some software and computational resources. So we've got plans to improve on what we've got out there, and we're actually currently doing some sequencing right now to get the hi-fi sequencing reads, which will make a much better resolution. Also trying to do some fluorescent in situ hybridization with tagged telomeres, so we'll be able to clarify chromosomes, because we think now we have 22 chromosomes. As I said, the PacBio hi-fi sequencing is on the machine right now. We're developing diagnostics for biosecurity, working with researchers from Canberra University and the Australian government. We're developing markers. Well, we are really concentrating efforts on looking at resistance genes across hosts and developing markers for resistance. And yeah, and then just understand the infection process. We will make a very good resolution reference genome for the host too. And so with that, I'll just thank all the collaborators and make some acknowledgements here. There have been a lot of people along the road who've been involved in this project. So yeah, thanks to everyone.
Thanks very much, Peri, for that. It's a journey, really, isn't it, to get to this point, and a lot of technical information there. So I can't actually see any questions on my screen. I'm hoping that isn't the result of some kind of technical snafu. Apologies if that is the case, but I do have a few myself. So I'm just going to start the ball rolling here. Now, I'm coming at this from completely non-expert background, my own background being far more in ecology and mycological ecology and no genetics. So forgive me for asking perhaps quite simplistic questions, but when someone like me, and of course the media is engaging with material like this, and we hear these things like GC content and huge genome and transposable elements, well, the first thing I tend to think is, do we understand the ramifications of this kind of information for disease expression or behavior? Like, can we link any of this to biology, or is the science just not there yet, and is it too complex?
Well, I mean, I hope that we can link it to, that's the whole point. So, I mean, I think the evolution of the genome is obviously under some selection pressure, and so, and because much of the reproduction of this particular rust is non-meiotic, so it's not going through sexual recombination. So we think there's a lot of random kind of mutations, I guess, that are happening due to just the millions and millions of spores that are being produced. And I guess that then it drives a selection against the host. So the transposable elements are likely to be driving a, yeah, so this is something we need to work out further, but there are other rusts, so there are other pathogens where you have these effector genes, so genes involved in parasitizing, which are actually located near telomeres or near sort of repeat regions, and that means that they are very easily changed, like they're maybe an insertion of a TE might kind of slightly change it, just so the virulence can change just randomly through these transposable elements. So I guess yes, but we don't really know.
So a classic example is when I've been talking to the media about, oh, it's got a huge genome. Well, of course, then they like to make the mental leap of, oh gosh, is that why it's so bad? But of course, that's not necessarily true. But I think what you're saying is there are actually elements of the genome that do lead into why this disease is so problematic and occupy so many hosts, and it is the fact that mutation is facilitated perhaps by these transposable elements, which themselves make a very large genome. So I guess the answer to these things is often yes, but it's complicated.
Yeah, I think yeah, that's probably true. I mean, I think my background is more in plants, and I've come into rusts, you know, working with Myrtle Rust, and it seems to me that, you know, they have a very complicated life cycle, these rust fungi. But yeah, the predominant life cycle seems to be clonal, and so yeah, the fact that they just produce masses and masses of these spores just allows for this variation to occur, and that is what's kind of driving this. And the lack of meiotic recombination means that they're not being fixed up in the process. So there's not, you know, the TEs are not being kind of managed sexually, you know, they're just kind of getting proliferating.
Yes, I see. So we do have one question here. Would it be worth getting a new Hi-C library to assist your assembly, perhaps starting with isolated nuclei?
Yeah, I think that our Hi-C library was pretty good. The quality looked pretty good, and I think probably there were issues more with the actual assembly. So when we're trying to, well, the repeats are also a problem, of course, but I think that I'm hopeful that we have Jana Sperschneider at ANU, who's developed her own software now that will be able to scaffold better once we just get a less chimeric genome. Yeah, I think there were a few things like we didn't know how many chromosomes we were starting with, and now I think we've got a better handle on that. And I think the Hi-C, no, I don't. At this point, I hope we don't need more. I think we've got enough. But it would be nice, I guess, just to get from one nuclei and one, yeah, it would be nice to be able to really differentiate that.
And I guess another question from me, perhaps again a little bit simplistic, but it seems to me there's this story of rapid technological improvement and this enormous effort that went into getting one genome. Of course, we've only got that from one strain, this pandemic strain. Has technology moved forward and expertise moved forward to the point where you can see in the next few years it might be much easier to get these genomes and we can get whole genomes from other strains? Or is it still the case that it's an enormous effort and probably will continue to be so for a while?
I think there's always going to be improvements to genomes, but I think what we will have is a reference genome, and I don't think everyone else needs to go ahead and do the whole thing like we did. The idea is that you have a species reference, and hopefully these biotypes will, you know, if the reference is good enough, the other biotypes, you can do sort of very low level sequencing and map to that, and then look for change along the way. So this is
you get bits of them and then, yeah, kind of map them back. And I guess I just had one last question as we come into 11:30. And you mentioned this CRISPR technology. Of course, even those of us who aren't really involved in genetics, this is on our radar and getting plenty of attention in the media. So it's great to see you're starting to work with that. So is the idea when you talk about testing protein functions with that technology that you knock out those proteins in a very specific fashion so that you can then test what they might do by their absence?
That's correct. Yeah.
Yes, good.
It's a bit of it's a bit hard in an organism like Myrtle Rust, but we did. It was a proof of concept to see that we could actually get the. So I'm probably not that clear anyway on that image, but the pink indicates, so that fluorescing pink indicates that it was in the hyphae. So it is targeting to the to the rust. So we're able to get it across the membrane and into the nucleus. That's the aim.
And I guess especially challenging if you're trying to work with it in the plant?
No, we were working with spores germinated on agar.
Okay, good. That sounds better. All right, excellent. Well, that's it for today. Thank you very much, Peri. Excellent to have you contribute to our Myrtle Rust webinar series. So for the audience still with us, a video of today's webinar and answers to questions will be made available on the Beyond Myrtle Rust website in a couple of days, and you should all receive a follow-up email pertaining to that. The next webinar will be next year, February the 10th. Same time, same place, and will be given by Dr. Mary Toom from the Ministry for Primary Industries in New Zealand. Now, this ministry handled the initial incursion response when a psidii arrived here. And Mary A's talk is titled Chasing Myrtle Rust in New Zealand, Biosecurity Response Diagnostics and Beyond. So I guess this really draws our attention to how many different sides of science, politics, activities are involved in managing a plant disease like this and understanding what's going on. So Merry Christmas, everyone. Until next year, I hope you all have an excellent break, and we'll see you in 2021.